Okay, so this is chapter 6, where we are going to talk about lipids. And so we'll talk about triglycerides, phospholipids, and sterols, focusing on cholesterol. And so, as usual, here is our list of learning objectives. Again, it's a long list that I will not read you, but by the end of the chapter, hopefully all of these make sense. Okay, so again, when we're talking about lipids, commonly we refer to lipids as fats and oils. All lipids will contain the same types of atoms, so they're going to be composed of carbon, hydrogen, and oxygen. Just like we saw with carbohydrates, but the ratios here will be different. Okay, lipids are a diverse class of molecules. There are many different types of lipids, very different length lipids. So triglycerides can be long chain, short chain, medium chain. So it's a very diverse class of molecules, but something that they all share in common is that they are insoluble in water, except phospholipids that have regions that are going to be what we call hydrophilic. They like water, but most lipids are what we call hydrophobic, water-fearing. So they're insoluble. They kind of push water away. They don't mix well with water. Lipids add flavor and texture to food. They also have over twice the amount of energy found in carbohydrates or in proteins. And so fats come in at 9 calories per gram, 9 kilocalories per gram. So again, energy source. We'll talk about that. Lipids also are important for insulating and cushioning organs. We're going to talk about that. And then lipids are also used to make hormones. Okay, so here is a table. Again, we have MyPlate over here, which you should be very familiar with by now. We have our key to MyPlate, right? And so half of your plate, just to remind you, should be composed of fruits and veggies. In the other half of your plate, we've got grains and then protein. And of course, dairy is also important. You can see that there's not really a spot for oils and others, and so, kind of sparingly, okay? And so foods rich in fats, okay? If we look at, for example, vegetable oil, and a serving of vegetable oil is usually 1 tablespoon. Or if we look at butter or margarine, again, same thing, 1 tablespoon. These are 100% fat, okay? All of the energy in these types of foods are from fat. There's no carbohydrate or protein in them. If we look at a medium-sized avocado, the same thing. Most of the energy in an avocado, over 80%, comes from fat. We looked at mixed nuts. Nuts are also high in fat, okay? Peanut butter, cheese, so on and so forth. When we come down here to like cereals like Special K, very low in fat. Fat-free milk, fat-free yogurt, very low. Okay, so again, foods rich in fats, and then foods that are kind of lower in fats. Okay, so again, kind of 3 types of lipid categories found in foods or made by our bodies. And so here are the 3 that we're going to talk about throughout this chapter. So the first is triglycerides, and we mentioned triglycerides with lipids back in chapter 1. So triglycerides are going to make up about 95% of the fat that we eat. This will be the fat that we use for energy, and this is the type of fat that we are going to store in specialized cells called adipose cells. Okay, we will get deeper into the structure of triglycerides, but here on this first slide, we tell you, triglycerides are fats and oils. They're made up of glycerol, 1 molecule of glycerol, and 3 fatty acids, and these fatty acids can vary. Glycerol is shown here on the left side of this molecule. I don't want you to worry too much about structure, but just to show you. And then the 3 fatty acids are 1, 2, 3. Okay, and again, we'll look a little bit closer at this structure in a couple of slides. Again, triglycerides are going to be used for energy, and they will also be stored for later use. We then have phospholipids, which we will talk about a little bit farther down in the chapter. Phospholipids are a little bit different. They're still made up of glycerol, so there's still this glycerol kind of backbone, and 2 fatty acids. And the third, instead of being a fatty acid attached to this first carbon, it is a phosphate group, which gives phospholipids a little bit of a different, not only structure, but functionality. So triglycerides are insoluble, they're water-fearing, they're hydrophobic, where phospholipids actually have 2 distinct regions. We call them amphipathic molecules because they have 2 chemically distinct and they behave differently. The fatty acid region is going to be hydrophobic, water-fearing, insoluble. But this phosphate group gives what we call polarity to the molecule, which makes it slightly, on this end at least, hydrophilic. And so again, we'll talk about that when we get into membrane structure and how phospholipids are involved there. Okay, so again, phospholipids are going to be the major component in cell membranes, and they are also precursors to neurotransmitters, which we won't really talk about. The last category of lipids are sterols, and again, our focus is going to be on cholesterol. Here is the structure. It's actually missing the top of it here, but it's 4 rings, 4 carbon-based rings fused together. And we'll talk about the role of cholesterol again a little bit later in the chapter. We'll talk about good versus bad cholesterol. And cholesterol does have an important role in our bodies. It is also part of cell membranes, and so it affects what we say the permeability of membranes. It's also a precursor to some hormones. So again, we will talk about phospholipids and sterols a little bit later in the chapter, but for now, we're going to start talking about triglycerides. Okay. So as we said on the previous slide, triglycerides are going to be fats and oils, and so pictured here looks like a vegetable oils over here, maybe canola oil. This looks a little bit darker. Maybe this is extra virgin olive oil. And then we've got some butter. Okay. All rich in triglycerides, or all, composed of triglycerides. So what is a triglyceride made of? Again, let's do structure. So a triglyceride is made up of 1 molecule of glycerol. Glycerol is a 3-carbon alcohol. It is considered to be the backbone of a triglyceride. And then again, we have attached to that glycerol 3 fatty acid molecules. And so fatty acids are going to be these long chains. They can be short, medium, or long chain, depending on how many carbons they are in length. And they're going to, though each carbon will be bonded to hydrogen atoms. And so carbon bonded to hydrogen is a very they're non-polar bonds, not to get into chemistry too much, but it makes these molecules insoluble in water. So they're not going to dissolve in water. Instead, they're going to kind of repulse water. And so, they're going to, if you had, a glass of water and you put oil into it, the oil will go to the top and sit on top. It's not going to mix, with the water. You can kind of think of salad dressing in that way. Okay, so let's see a picture. So I showed you a picture a few slides ago, but let's look at it again in a little bit more detail. So again, this is a triglyceride. Here is glycerol. So again, it's a 3-carbon, we call it a 3-carbon alcohol because it has these OH groups on it. An OH group is called an alcohol group. Okay, so this is glycerol. So there's 1 component to the triglyceride. And if we look up here in this figure, it's this blue part. And then attached to the blue part are these 3, orange squiggly lines. So what are these? This is the fatty acid. And so a fatty acid is going to have on one end, the end that attaches to the glycerol, it will have a carboxylic acid group, or we can just call it an acid group. So it's a carbon double bonded to an oxygen and then single bonded to an OH group. That's an acid group. Okay, this acid group is going to join with the glycerol at the hydroxyl group. And so it's going to happen here, and then a second fatty acid will add here, and a third fatty acid will add here, and you end up with a triglyceride, so 3 fatty acids. So here's the acid end, and then we have this, long stretch of carbons and hydrogens, and the number of carbons varies, and the number of hydrogens vary, and the type of bonds between the carbons. Sometimes they're single, sometimes they're double. So we're going to get into that as well. So we have the acid end, and then we have what's called the omega end. That's the opposite end. Okay, so again, triglyceride is 3 fatty acids attached to a glycerol molecule. Okay, so like I said, the fatty acid lengths can vary. So the length of that carbon chain, it could be short. So any fatty acid less than 6 carbons long is considered to be a short chain fatty acid. That's what we call them. 6 to 10 carbons long is considered to be a medium chain. And then longer than 12 will be a long chain fatty acid. Okay, so there's one way in which triglycerides vary, is the length of the carbon chain of their fatty acids. The second way is their level of saturation, and so that's what's pictured on this slide. The term saturation, this refers to how many hydrogen atoms each carbon atom is attached to. Okay, so if I look at this top, I ignore the acid end, but if I look at just the carbon chain here, right down to the omega end, you will see that every bond in between the carbon atoms is it's a single line. So we call those single bonds. And so in this top fatty acid, each carbon is bonded to the maximum amount of hydrogens. We know that because there's no double bonds present in the molecule. So this would be called a saturated fatty acid. The carbons are saturated with hydrogen atoms. We can't add any more, right? They're at their maximum capacity. On the flip side, below, this is an example of an unsaturated fatty acid. And so again, we have our acid end, we have our hydrocarbon, carbon and hydrogen chain. And look right here, the carbons right here are not attached by a single bond. Instead, they're going to be attached by a double bond. So that's these two lines here represent a double bond, which is 2 pairs of electrons being shared if you've taken chemistry. In this case, with this double bond, if we take a look at the hydrogen atoms, and then we take a look at the longer carbon chains, the hydrogen atoms are on the same side of the double bond. Okay, and so we say that this is in the cis configuration. So we're going to talk about cis versus trans unsaturated fatty acids as well. Okay, so saturated, single bonds, unsaturated, double bonds. And then lastly, because of bonding, fatty acids will also differ in their shape. So if they are saturated fatty acids, they're going to be straight. And if they're cis unsaturated fatty acids, they're going to be bent, or there will be a kink in them. If they're trans fats, you've probably heard that term trans fats. So those are trans unsaturated fatty acids. They tend to be straighter. They tend to behave like saturated fats. Okay, and so again, we're going to get into that cis and trans in just a second. But again, you should know that triglycerides do differ in shape. So we've got straight chain, and then we've got bent forms. And bent comes from double bonds. Okay, so I want to quickly take you through. So we said saturated and unsaturated. You may have also heard of MUFAs and PUFAs. So we can have monounsaturated fatty acids and polyunsaturated fatty acids. So let's take a look at these 3. So again, saturated, all single bonds. Straight chain. Notice the shape of the molecule, very straight, very linear. Monounsaturated. Mono means 1. And so again, if we look at our hydrocarbon chain, carbon and hydrogen chain, we find that there's just 1 double bond here. Okay, notice the shape of this molecule. Instead of being straight, there is a kink in it, right? A bend. That bend is going to cause this type of fat to behave a little bit differently. And then lastly, PUFAs, polyunsaturated, more than 1 double bond. Okay, so we can see a double bond here, which creates this little kink. And then we have another double bond here, which creates another little kink. Okay, so those are what those terms stand for. Okay, so let's talk about structure and what that means when we look at the type of fat. Okay, so again, here is the same picture again for us. So here's glycerol, our backbone of our fatty triglyceride. And then we have 1, 2, 3 fatty acids, and in this case, there's all single bonds between the carbons of all 3 fatty acid chains. So this is a saturated fat, okay? This type of configuration, which is shown here again, would be found in hard fats, so like butter, okay? If we look at butter, if we were to look at the composition of the triglycerides, we would find all saturated fatty acids, okay? So all of the fatty acids have single bonds between all of the carbon pairs, which again means that this fat is saturated with hydrogens. We can't add any more hydrogens in. The maximum number is there bonding to carbons. Okay, and if you haven't taken chemistry or bio, just as a little maybe reminder, or just to kind of let you know, carbon can bond to 4 things and only 4 things. Hydrogen can bond to 1 atom and only 1 other atom. And so that's why you see each of these carbons, any carbon, you can pick any carbon out on this page, and each carbon is bonded 4 times. That's because carbon has 4 unpaired valence electrons, and so it can bond 4 times. Okay, just to give you a little bit of context. And then hydrogen, you'll notice each hydrogen anywhere on this page is only bonded to once, to 1 other atom, only a single covalent bond. And that's because hydrogen has 1 valence electron. Okay. Let's look at oxygen just because we're doing this. So we can look at the oxygens of glycerol, or we can look at the oxygen of the acid group. You'll notice that oxygen is always bonded twice, and that's because oxygen has 2 lone unpaired valence electrons. So oxygen can only bond twice. Okay. Okay, so back to saturated versus unsaturated fats. So saturated fats are found in solid fats like butter, lard, Crisco. Okay. Unsaturated fats are going to be found in softer liquid type fats. Okay, so we call these unsaturated, and these are going to be oils. Okay, oils like vegetable oil, canola oil, olive oil, avocado oil. Those are all unsaturated fats. And again, if we were to look at the hydrocarbon chains of the triglycerides in these oils, you'll notice that they're all bent and kinked, and that's because of double bonding. Okay, so some of these, this one looks like a polyunsaturated fat. This one looks like maybe just a monounsaturated fat. Okay, so the level of double bonding is also going to affect the shape of that molecule. The more double bonds, the more kinks or bends you're going to have in that molecule. So you can see in a saturated fat how the fatty acids, because they're straight, they can compact, right? They're going to be really tight together. Whereas in an oil, those unsaturated fats with kinks, there's spaces in the molecule. And that's why we get a different texture, right? We don't have a hard fat, we have an oil. So butter, lard, hard fats are full of saturated triglycerides, and liquid fats, oils, are going to be full of unsaturated fats. Okay, there's the key difference. That is why they're in different, states, solid or liquid. Okay, and I want to quickly bring us back to cis versus trans fats. This is pretty important. So both cis and trans are unsaturated. Okay, but how the double bond sits in the molecule is going to determine if it is a cis or a trans configuration, and that will determine the shape of the molecule. Okay, so let's look. In this figure right here on the left, we have a cis unsaturated fat. And so here's the double bond right here. And again, you'll notice that the hydrogens are on the same side. They're in the cis configuration. Cis means same. They're on the same side of the double bond. Okay, where's my pointer? There we go. A double bond, when a double bond is between those 2 carbon atoms, the atoms around that double bond can't move freely. They're kind of locked in plane. Okay, so they're locked in that cis configuration. And again, we get a nice bend in the molecule. Okay, so this particular fatty acid right here is called oleic acid. So again, cis form, hydrogens are on the same side of that double bond. We get a kink or a bend in the molecule. unprocessed fatty acids like nut oils, seed oils, which includes, vegetable oils, things like that. Those are all going to be in this cis configuration. Trans fats, you may have heard of, you may have seen them on labels. Trans fats have actually been banned in the United States. They have been found to be very unhealthy. We're going to come back to this in just a little bit. We'll actually talk about how they're made and everything. So let's look at the trans form. What does it mean? So there's still a double bond, so it's still considered to be unsaturated. Okay, but when we look at the configuration of this double bond, the hydrogen atoms are across from each other instead of being on the same side. So we say that they are in trans. And so instead of creating a bend, we end up with a straight linear molecule. And so trans fats behave like saturated fats. Okay? They look like saturated fats. Okay, let's get into it a little bit further. Okay, so first off, trans fats rarely occur in nature, but they do. Okay, so if you're looking at a food label currently in the United States and you see trans fats on there, and if there is any, those are naturally occurring trans fats. And they're actually very different from chemically produced trans fats. So there's a difference there. So again, trans fats rarely occur in nature. So most unsaturated fats are going to be cis. Okay. Trans fats usually, again, are made chemically. So what we do is we take a nice, healthy, unsaturated fat, and we do this process called hydrogenation. Okay, so again, we take a nice, healthy, cis, unsaturated vegetable oil, and we do this thing called hydrogenation, where we, there's on the next slide, there's actually a picture of this. We take and we add hydrogens to the fat. And when we do that, again, it changes the configuration, and it creates this trans configuration in which the fat now, instead of having this nice bent form, is going to be linear, straight, and rigid, just like a saturated fat. Okay, we used to, in the United States, again, they are banned, but we used to use trans fats in margarines, shortenings, and ultra-processed foods. Okay, we did this, again, they were added to food to extend the shelf life and for cost. So, vegetable oils like corn oil is pretty cheap, right? And to make the corn oil more shelf stable, this process was done and it was added to foods, and then that would extend their shelf life, and it's cheap. Okay. Trans fats, again, have been found to be really bad. They are very detrimental to health. Number one, they change the way in which our cell membranes function, so at the molecular level they're pretty bad. They also reduce bad cholesterol from the blood. Okay, so that's a process that happens. Trans fats have been shown to decrease that. And that will lead to atherosclerotic plaques and cardiovascular disease, which are two things that we're going to talk about towards the end of the semester. So trans fats, they've been shown to raise LDL levels, which are bad cholesterol. And again, we're going to come back and talk about LDL towards the end of the chapter. Again, I want to just hit this home. Scientific research was done. We figured out that trans fats are not good. And so again, in 2018, we officially banned trans fats. Some companies had a little bit longer to get them removed from food, but the science supported this, and it was done. Okay, so when you see it on food labels now, zero trans fats, that's a marketing ploy. This process doesn't happen anymore. Companies are not allowed to put trans fats into foods. And again, there was scientific, evidence-based reasons for that. Okay, I want to just briefly mention this process of hydrogenation. So hydrogenation, again, we do this in a machine where hydrogen gas is added under pressure. It forms tiny bubbles at the base of this thing called the agitator. There's a nickel catalyst in there. So it's a process. Oil temperature increases once the hydrogenation process begins, so it's an ectothermal reaction, and hydrogen gas is used here. So what are we doing? We're taking a good, healthy, unsaturated oil, like a vegetable oil, putting it in this chamber, and we're going to do this hydrogenation process where we add hydrogen gas. And again, it's going to convert these nice, healthy fats to more solid form by adding hydrogens to the molecule. It used to be used to create margarine from oils. And this process of producing trans fatty acids, this is called partial hydrogenation. So that's really important. Partial hydrogenation led to trans fats. If we do a full hydrogenation, it just leads to saturated fats, which are still not great. We'll talk about what's good and what's healthy and what's unhealthy in a couple of slides. But again, partial hydrogenation led to trans fats, and trans fats have actually been shown to be possibly worse than saturated fats. Okay. Do we still do hydrogenation? Yes. But instead of partial hydrogenation leading to trans fats, usually it's a full hydrogenation leading to saturated fats, and those fats are still used in ultra-processed foods. So still not great, but again, at least we've gotten rid of trans fats. Okay, and just to show you again, unsaturated, saturated, and trans. So unsaturated fats, you can see this picture of the avocado here on the left. If we look at the chemical structure, look, nice bend in that molecule, a nice kink. And again, unsaturated fats, we'll see, they're sometimes called good fats. They can be found in things like nuts, avocados, and other vegetables, vegetable oils, avocado oil. Those are all full of unsaturated fats. Okay, and again, we'll talk about the good, the bad, and the ugly in a little bit. We have saturated fats that's shown here in this marbled meat, right? This looks like bacon. These are very linear, straight, and rigid. These fats are found mostly in animal products. Again, it's recommended, we'll talk about this in a little bit, that people, you don't have to consume no saturated fats, but you should try to limit your consumption. And then trans fats, which we said have been banned. They are considered to be unsaturated, but they've been, if they're processed, right, they can be found naturally as well. But if they're the processed form, they're going to be partially hydrogenated, again, to extend their shelf life. And we've found that these are really bad for cardiovascular disease because they elevate LDL. So we'll learn LDL stands for low density lipoprotein, which is bad cholesterol. Okay, we're almost wrapped up here. We've got about 3 slides left. Our focus for these 3 slides is going to be on essential fatty acids. So there are 2 fatty acids, PUFAs, polyunsaturated fatty acids, that are called essential fatty acids because our bodies can't make them and we have to obtain them from our diet. So we have linoleic acid, which is also known as omega-6 fatty acid. And then we have alpha linolenic acid, which is also known as omega-3 fatty acid. So omega-6 fatty acid, this linoleic acid, can be found in vegetable oils, including seed oils like soybean oil, corn oil, canola oil, sunflower oil, safflower oil. Those are all examples of seed oils. And then we can also find omega-3 fatty acids in nut oils. Okay. And then we have the omega-3 fatty acids, which can be found in vegetable oils as well as fish and fish oils. Specifically, fatty fish are going to be high in omega-3 fatty acids. Other things too, like chia seeds, are really high in omega-3 fatty acids. In the United States, the standard American diet, we tend to get high levels of omega-6 fatty acids and not enough omega-3 fatty acids. And so we'll talk about this again a little bit later in the chapter. But the ratio of omega-6 to omega-3 is really important And, Americans tend to not get the proper ratio. So the ratio is, somewhere around a 4-to-1 or lower is good. And Americans get like a 15-to-20-to-1. And so again, our ratio is off, which is not a good thing. Why do we get so much omega-6 fatty acids compared to omega-3s? And so a lot of the healthy vegetable oils, including seed oils, they get processed. And so we talked about hydrogenation in the previous slide. And so instead of being partially hydrogenated, they actually get fully hydrogenated. That's not banned. So when we do that, we take these nice healthy oils, including seed oils. When we hydrogenate them, we add hydrogens, and now those seed oils are saturated. And so that's bad. That is found, those types of processed oils are found in ultra-processed foods. Why? Because vegetable oils tend to be cheap. We can do this process called full hydrogenation, and that increases the shelf life of ultra-processed foods. Okay, so it makes them cheaper to produce, and they last longer. And so Americans tend to eat a lot of ultra-processed foods, and so we get a lot of omega-6 fatty acids. So in reality, seed oils are not inherently bad. We just overconsume them, and again, a lot of that is through ultra-processed foods. Okay. And so again, it's not the seed oil, it's the processing of the seed oil, right? It's also, in ultra-processed foods, you also have, again, usually high amounts of sugar, high amounts of sodium, high amounts of calories, low amounts of fiber, vitamins, and minerals. And so overall, they tend to be energy dense and nutrient poor. Okay, so we overconsume omega-6 fatty acids, and we don't get enough omega-3 fatty acids. And again, we'll touch on that after. Okay, so why do we need essential fatty acids? Why do we need omega-6s and omega-3s? One important function, we will touch on a few others a little bit later in the chapter, but one important function is because we need them to make these biological compounds called eicosanoids. Some examples of eicosanoids are things like prostaglandins and leukotrienes. What are these types of things involved in? They're involved in regulating gastrointestinal mobility. They're involved in regulating blood clotting, contraction and dilation of blood vessels, so blood pressure. They're involved in regulating our immune system and inflammation. They also reduce the negative impacts of saturated and trans fats. Okay, so very, very important fats in our diet. Okay, and here's just an example. So here are our essential fatty acids. So we've got omega-3s, we've got omega-6s. Again, omega-3s are known as alpha-linoleic acid, and omega-6 fatty acids are known as linoleic acid. And both of these produce eicosanoids, okay, through a pathway. Omega-3 fatty acids also produce what's called EPA and DHA, which we will touch on in a little bit later in the chapter. Okay, so that is the end of part 1 of chapter 6.